Good afternoon, students. Hope you all are doing well and staying safe. In this video, what I want to do is start moving us in the direction of putting together the different pieces that we've learned about in lessons 1 through 5. If you haven't completed assignments 1 through 5 up to this point, please go back and do those before you continue with this video. If you have, we're going to start with just a quick recap. So the question that we are trying to answer here is, how does a mutated CFTR channel protein cause a person's mucus to become thick and sticky and build up inside of their airway? Remember, we started this whole thing by me telling you about this genetic disease, this autosomal recessive genetic disease called cystic fibrosis, which mutates this thing called the CFTR channel protein. And people with this genetic disease, they develop this very thick mucus inside their airways that we see right here. And not only does that make it hard to breathe, but it also can lead to very high rates of lung infections, which can be very fatal. So before I get into the focus of what you've done the last few assignments, just a quick recap from about a week and a half ago. So we started this by trying to understand, how does the CFTR gene control the CFTR channel protein? And you learned about two processes, transcription and translation, which I have put over here on the left-hand side. And so just to be clear, when we're talking about this gene, this section of DNA on chromosome 7, when a cell wants to create the CFTR channel protein, it's going to transcribe that gene into RNA, which is a single-stranded nucleic acid. And then a ribosome is going to come in and translate in groups of 3. Every 3 nucleotides will lead to 1 amino acid. And the chain of amino acids we refer to as a protein. So that gene, the CFTR gene, gets transcribed and translated into protein. And in this case, we're talking about this channel protein. Now, you also learned about, if a mutation occurs, right? If a mutation occurs up here, it changes the DNA sequence such that it'll change the RNA sequence, and it might change the amino acid in the protein. All of those things will lead to a mutated CFTR channel. And just visually, what you can see here is there's a slightly different shape to these two proteins, right? This is the normal CFTR channel protein, and this is the mutated CFTR channel protein that was caused by a change in the gene sequence. So that's where we started assignments, I believe, 1 through 4. Now, the last few assignments, you've learned about 3 other things. If we have a changed CFTR channel protein, how does that lead to this? Well, so we started by thinking about what the mucociliary system is. That's where we find this channel protein, is on the membrane of cells involved in the mucociliary system. So you started by learning about the mucociliary system, which is over here, right? And its job is, especially in your airway, right? Your airway right here, and your nasal cavity, breathing through your mouth down to your throat, right? All of this is coated in a mucus, and it traps things like pollutants, but also traps things like viruses and bacteria, which can cause infection. And so you learned about how the mucociliary system is really an important line of defense, because we're breathing in and out all the time, and that mucus is like a net that prevents us in a lot of ways from getting sick. So you learned about the purpose of the mucociliary system. And then you started learning about membranes, right? Because membranes are where these channel proteins are found. So if we were to zoom in over here on the top right on one portion of this airway cell, we would find on the outer barrier of those cells a membrane, right? And if you played that little game, that online game, you learned about a couple of different things that we find on the membrane. So if you take a look down here with me, you learned about how the membrane has two layers. We call it a phospholipid bilayer. If you notice, right, this is composed of two layers, and it has what are known as heads and tails. And you learned about how the tails, they repel water, and the heads, they attract water. And a membrane forms these dual layers of these molecules, these phospholipid molecules. But that's not all that you find on the membrane. You find a lot of times these proteins, among other things, that are kind of lodged inside of this membrane. Because at the end of the day, right, this membrane is trying to protect the inside of the cell. So if we take this to be the inside of the cell and this to be the outside of the cell, a cell doesn't want to just let anything in, and it doesn't want to allow everything to go out, right? It wants to create kind of a stable internal environment to be able to survive. But you learn that there are some substances that can actually just pass straight through the membrane, right? Things like oxygen or carbon dioxide, they can just pass through pretty easily. But there are some substances that they need a protein to help move themselves from one side to the other. So I have two examples here, right? Two different shaped proteins that are involved in something called facilitated diffusion. And the reason why they call it that is that it's helping diffuse a substance from one side of a membrane to the other. And just like regular diffusion, there's only one direction that these molecules can go, and that is the direction determined by the amount of that substance on both sides. So if you notice, there is a high amount of these triangles or these spheres, and there is a low amount of them on the other side. And so we say that diffusion can only occur from a high level of that substance to a low level, right? And it's going to continue to do that until there is a balance achieved on both sides of that membrane, right? And this occurs very readily. It doesn't need energy. It's not difficult for the cell, as long as it has that protein that specifically helps that substance diffuse, okay? Now, you also have the other type of protein, because you might be thinking, well, what if you want to go the other direction? Well, so in this case, if you look at the squares, right? In the squares, on this side, you have a low amount of the squares and a high amount of the squares on the bottom. But yet you see the squares traveling from low to high, and that's possible, but you need some energy. And we know that energy that's formed in cellular respiration is ATP. And you need energy to be able to pump that from one side to the other, okay? When we think about CFTR, a CFTR channel falls under this category right here. The substance that it's helping move from high to low, look at these chloride ions, is within the airway cells. Chloride ions can move from high concentration to low through this channel protein, and that's facilitated diffusion. Okay, now the other thing that you learned about is osmosis. And osmosis is the diffusion of water across the membrane. Now, water is one of those substances that it can move across the membrane by itself without a protein helping it. It moves kind of slow. However, there are some proteins that you saw in the online game called aquaporins that function just like these right here, and they can allow water to go through the membrane very fast. And so this is just simply the diffusion or facilitated diffusion of water. And that happens in a particular direction. You saw a similar figure as I've put on this slide that looks like a U-tube, a U-shaped tube. And it's said that water, it moves from an area where there is low solute concentration to an area where there is high solute concentration. Remember, solutes are what is dissolved inside of the water, which is called the solvent, right? So we see water can pass through the membrane of this U-tube to the other side. So now we're going to get equal amounts of solute on both sides, the concentrations of those solutes. Okay, so that was a recap of everything that you've done so far. Now what we need to do is we have to be able to connect, after we have our channel protein, if it's mutated, how can we take into account substances moving across the membrane and osmosis and the mucociliary system to all arrive at an answer of what causes the mucus to become thick and sticky and build up inside of their airway. And that's what I'm going to talk about next. Okay, so now that we've done a review of the key parts of the last few assignments, we're going to take a little bit more of an in-depth look at what's happening inside of the airway when you have a mutated CFTR channel. So once again, we're talking about the mucociliary system. So just to get acclimated to the model that I'm going to draw right here. So the mucociliary system is composed of the mucus that's found inside of the airway, right? So the airway is this tube. You have specifically things like the trachea, which goes down into the lungs and branches off. And then you have your nasal passageway, you have your mouth, and then ultimately connecting at the throat. And so the mucus, like we said before, it catches things as you breathe in and breathe out. And the mucus, once it's caught those things, normally, using these little brush structures called cilia, it actually starts to sweep up the mucus, like we see in this diagram, to your throat. Or if you're in your nasal passageway, it sweeps it to your throat as well. And once it gets to your throat, it either will be coughed out, sneezed out, or will be swallowed, and anything that was contained in that mucus will be destroyed in the stomach. And so it's really this elegant system of defending and destroying against pollutants and viruses and bacteria. So what we're going to do is we're going to zoom into a few of the cells that you would find inside of this airway. So just for some perspective, so if you see down here, looking right here, they zoom in on a small portion of the airway, specifically some of the branches inside of the lungs. And they give you these two zoomed in images, a healthy airway and one that has cystic fibrosis. So in this figure above, right, that corresponding box would be right around here, okay? So I want us to see the connection there. But what we're going to do is we're going to take even a closer look at this boundary, all right, in both of those. Now, first, we're going to look at just a healthy airway, and then we'll get into one with cystic fibrosis. But remember that although this looks like just one continuous tube, if we zoom in, let me draw a little dotted line here for you. If we zoom into that area, what you would actually find are individual cells. This is made up of individual cells that compose the mucociliary system. It's at that boundary of the airway and the mucus, right? That's the mucociliary system. And so if you look right here, we have a couple of components. So these right here, this is an airway cell. And like any cell, it has a nucleus. Now, if you notice, some of these, they have cilia, which are those brush structures that help push mucus up. In the context of what we're zooming in, this would be this area. This would be inside the airway, right? Meaning that airflow would be going up and down. Airflow would be going up and down as we breathe in and out. Now, what we would also see is on the other side, so this is not inside the airway, you would see blood vessels. Blood vessels, which are part of our circulatory system, they help carry nutrients to the cells as well as help them get rid of waste. And so we'd see blood vessels kind of partnered up with these cells to provide the nutrients that they need, like glucose or a couple of other ones that we'll talk about in a moment. Okay, so one other thing that we need to know here, I'm going to use yellow to indicate the mucus. So if this is inside of our airway, then we would expect to see, normally, a layer of mucus right there, right? So everything that would be here would be a layer of mucus. Okay. And just for reference, so mucus, this cilia is going to push that mucus back up the airway so it can be either sneezed out, coughed out, or swallowed. Okay, so that's going to be the mucus flow. Now, inside of that mucus, again, we would expect to find a couple different things. We'd expect to find, we're going to use this to represent pollution, so air pollution. We'd also expect to find probably things like bacteria, okay, and we'd probably expect to find some viruses. So scattered about here, we'd expect to find all of these types of either pollutants, viruses, or bacteria. Okay, that gets swept up through the mucociliary system to the throat. Okay. Now, if you notice, in our cells, I have 4 cells here, right? 1, 2, 3, and 4. Every one of those cells has a nucleus. However, not all of those cells have cilia, and that's because there's 2 distinct types of cells inside of your airway. The first type of cell that you see right here that has cilia on it, we call that a ciliated cell. However, the cells that don't have cilia, like this one, we call that a goblet cell. And we're going to talk about goblet cells a little bit later. But just so you know, when I'm referring to a ciliated cell, those are the ones with the little brush structures on them, and goblet cells are the ones without. Okay. Now we're going to start pulling in things like membranes and those channel proteins and osmosis. So to kind of further illustrate that on this model, keep in mind, right, when we think about membranes, let's take this bottom goblet cell. If I were to zoom in right here, what would we expect to find? Well, we'd expect to find that phospholipid bilayer. Just for simplicity, right, I've just indicated the boundary with a line, but in reality, you would expect to find a phospholipid bilayer, right? A membrane is surrounding each one of these cells. And embedded in those, we'd expect to find a couple of different things. The first thing that we would expect to find is on the membrane of the ciliated cells, right? Like this top one or the bottom one, you're going to find our CFTR channel protein. So what I'm going to do is I'm going to use green here. So on the side that's adjacent to the mucus, I'm going to draw two CFTR channels. One there and one there. And then down here as well. Now, CFTR, it's not found on the goblet cells, right? But it's found only on those ciliated cells. So I'm going to draw a label here. So remember, this is our CFTR channel. Okay. Now, the other thing that we're going to find is on the opposite side of this ciliated cell, you're going to find something called a chloride transporter. So these are, again, just on the ciliated cell. This is called a chloride transporter, and I'll talk about what those are in a moment. And the last thing that we're going to find here is on the membrane of every single cell, we're going to find aquaporins. And if you remember what aquaporins are, they're channel proteins that help facilitate the diffusion of water very fast. And so osmosis can occur at a very fast rate. So I'm going to label one of these. This is an aquaporin. Okay, so we have our 4 cells, ciliated and goblet cells, and we have a membrane surrounding each one of those. And we have on the membranes, we've got CFTR channel proteins, we have aquaporins, and we have chloride transporters. So we're going to start to tie this together. And to do that, we need to know what's contained across this entire system. So the first thing that's going to be super important here is we need to know that water is found everywhere in this system. So to illustrate that, what I'm going to do is I'm just going to put a bunch of blue dots all over the place. Now, water, it is often called the universal solvent. And that's just because, at least in living organisms, within cells, within the mucus, all of these different things, water is dissolving everything, right? So if you remember, solvents are what dissolve solutes, things like sugar or sodium, or like we'll talk about, chloride. Okay, so we have water all over. Okay, and it's in our mucus as well. Now, the other thing that we're going to find is that whenever we eat or drink certain things, you actually extract chloride. Chloride is an ion, and I'm going to indicate that with Cl-. And so chloride is actually floating through your blood a lot. It's something you might have heard of the name electrolytes. It's an example of an electrolyte. And so I'm just going to indicate right here, this is a chloride ion. And then I forgot to label, right, our little blue dots. That's just water. Okay. All right. So we have water, we have chloride, and then we have our goblet cells. Our goblet cells produce the other component of mucus, And so inside of the goblet cell, there is a gene that codes for a protein called mucin. And so what this will produce is something called mucin. Okay. And so when this protein is produced, it gets sent out into the mucus, and that's what helps keep the mucus layer going, right? It's this combination of water and mucin. Okay, that's what gives it its kind of sticky property. Okay, so before we go further, just a quick note that I want you to make up here is that water, this is the solvent, and chloride or chloride ions are what is dissolved inside of water. So we have in the blood, we have chloride as the solute and water as the solvent. Now there are other components that we don't need to know for right now, but that's going to be useful when we think about osmosis, right? We're going to think about osmosis in a moment. Now, to tell you a little bit more about these membrane proteins that you've drawn, you have a chloride transporter protein and a CFTR channel protein on your ciliated cells. So, normally, these chloride transporter proteins, we'll take just this top cell right here. What they will do is they'll take a chloride ion and they'll actually pump it into the ciliated cell. So you'll get chloride here. And on the other side, chloride can go through this channel protein and into the mucus. So another component that you'll expect to see inside of your mucus is actually chloride, right? Because it gets ultimately into the mucus after it's gone through the chloride transporters and the CFTR channel protein. So you kind of have this system of chloride movement. Okay, now remember, we're talking about a healthy situation, right? So chloride is getting pumped in, and then it's getting facilitated out. So now, considering that, we're going to think about in a situation involving CF or cystic fibrosis, how does osmosis play into this? So to answer that question, what I've done here is I've zoomed in on one ciliated cell. Right, remember there's ciliated cells and goblet cells, and I've divided this image into halves. So the first half is one ciliated cell that is healthy, that is, it doesn't have cystic fibrosis, and then in the other one, we have a cystic fibrosis ciliated cell. I have all the components already on here. We have our blood vessel, we have chloride, we have water, we have the bacteria and viruses caught in the mucus, and we have our nucleus. So what I want us to do is let's look at the healthy ciliated cell to think about how osmosis plays into this. So, like I mentioned earlier, when a cell is healthy, it will pump chloride into the cell. Right? It'll pump chloride into the cell. And as that happens, because we're eating, we're drinking things that have chloride in them, you'll start to see, right, chloride is going to, the amount of chloride in the cell is going to start to grow. But on the other side, remember there's a way for the chloride to get out. We have these CFTR channel proteins, right? So we have our transporter over here, and our CFTR channel over here. And that allows chloride to leave the cell and get into the mucus, just like that. So you have a way in and you have a way out. Now, just a quick note, remember that these proteins, like CFTR channels, right, they came from a CFTR gene, right? Like I've put in the nucleus here, there's a particular section that gets turned into, through transcription and translation, it turns into these channel proteins. So don't forget about that, because that's going to be important for the next part, is that's based on the CFTR gene, which allows that chloride to pass through. Okay. Now, in the case of a cystic fibrosis ciliated cell, the CFTR gene is mutated, right? So you're still going to get transcription and translation, and that protein is still going to find its way to the membrane. However, it's going to be shifted. It's going to be changed in shape. So it's going to be there, but it's actually going to be closed, right? It's not going to be functioning. So I'm going to indicate that with an X. So that's a mutated CFTR channel. Now, nothing has happened to the chloride transporter, so what's going to happen is you're going to get chloride coming into the cell, and chloride is going to start to build up like normal. However, there's no way for it to escape, right, on the other side. So you're going to get chloride building up like crazy inside of this ciliated cell. Now, why is that dangerous? Well, because remember that chloride, or chloride ions, those are the solute in this system that are dissolved in water, which is the solvent. Now, what we're starting to see here is that there is going to be an imbalance of chloride inside the cell versus outside the cell in the mucus, because look, there's no chloride that's entered the mucus right here. So what's going to start to happen is, since chloride can't make its way out, you're going to get a situation where you have an imbalance in solute. And if you remember from the osmosis video, we call a situation where there is a lot of solute, we call that hypertonic. And when there is a low amount, we call that hypotonic. And water is going to move in a specific direction in order to restore balance, or isotonic situations. And so remember the aquaporin, which has been right here the whole time, right? This aquaporin is actually going to facilitate osmosis to occur very fast. And so what's going to happen is that all of these water molecules, they're going to start to diffuse a lot into the cell. And so what you're going to start to see as that happens, you're going to see water start to disappear from the mucus, right? Because they're transporting into the cell, right? Because again, its goal is it wants to restore a situation where you have equal concentrations of solute on both sides. So you're going to get a ton of water in here and a lot less water in the mucus. Now, a question that I often get asked by students about is about the chloride transporter. Because if you think about the chloride transporter, wouldn't it be better if the body could just stop pumping chloride into those cells by maybe shutting down the chloride transporter? Like, why does it continue to pump chloride even though the concentration is very high? Well, remember in the membrane online game that you played, right? There are some proteins on the membrane that can pump from low to high, and chloride transporters are one of those proteins. They can pump chloride from low to high concentrations. Now that requires energy, requires ATP, but in other words, the chloride transporter is going to continue to pump chloride even though the concentration is higher inside of the cell than it is in the blood vessel. CFTR, unfortunately, though, can only move chloride from high to low. It only facilitates diffusion. Okay. So, to recap, we have a buildup of chloride inside of a cystic fibrosis ciliated cell that leads to osmosis of water through the aquaporin into the cell. When normally you have a situation where there is an isotonic concentration of chloride in the mucus and in the ciliated cell, so you don't get any net movement of water into or out of the cell. Okay, now remember the question we're trying to answer is how does that lead to thick, sticky mucus that builds up inside of the airway and causes all of these issues? Well, so if you think about this situation down here with the cystic fibrosis cell, when you remove water from anything, right, it becomes dry. What was the other component of mucus? Well, it was that mucin. It's that sticky substance that normally is kind of dissolved a little bit in the water. But when you take out all of that water, this mucus down here gets very, very, very thick, such that the cilia, right, when they try to sweep the mucus, because remember their job is they need to sweep the mucus up the airway, they actually can't do it because it's so thick that they're not strong enough to be able to sweep up that mucus. And so it stays there along with everything that's caught inside of that mucus. Whereas normally up here we still have, right, we have a ready mucus flow, but down here that flow is blocked because it's too thick. Okay, so not only does that mucus become thicker because the water is leaving the mucus, and because of that the cilia can't push it up the airway. Remember the other type of cell that's in the airway is the goblet cell. And the goblet cell, I mentioned, it produces the major substance of mucus, which is mucin, right? So mucin, which is being produced by these cells, mucin is functioning perfectly fine. And it's going to continue to be produced and released into the mucus. So if you think about this, not only is the mucus stuck there, causing issues with sweeping it out, but then you have mucin continually being added to that mucus. So it's almost like a traffic jam on the 405 where you have this continual buildup of mucus. And so what you tend to see over time is that this mucus layer gets thicker and thicker and thicker until, remember that the airway is getting constricted. And so we start to see the phenomenon of it's hard to breathe, and the stuff that's stuck in there starts to cause issues. Because even though it's trapped, does not mean that it's killed. So that bacteria and the viruses are going to start to divide and cause infections in the lungs. And so really that's what we're looking at when we think about the cystic fibrosis transmembrane conductance regulator, right, CFTR. When it's mutated, it causes this buildup of thick and sticky mucus inside of the airway, which again was where we started, right? We have now explained how we see this phenomenon of this mucus becoming thicker and sticky and constricting airflow as it becomes built up inside of the airway. That's what we just modeled using these cells. So I hope that was helpful. Please don't hesitate to reach out to address any questions. Once you have finished this assignment, you can go ahead and take our quiz. That's going to be based on assignments 1 through 8. And then our last piece of this is we're going to start to talk about, knowing this mechanism, how can we start to think about therapeutic treatments or even cures for someone with cystic fibrosis. That's really the last piece of this, is understanding a disease is one thing, but then also being able to use that understanding to solve or treat or cure that disease is kind of the last big step. I hope you all are doing well. Talk to you soon.